Good morning, everyone. We're back for a third day of the Global Summer School. And today we will welcome UPO from Novara, Italy. So I give the word to Francesca Boccafoschi, who will kick us off today.
Okay, here I am. I'm sorry, at the very last second I had trouble with the connection, but here I am again. So I would like to say hello to everyone who is connected with us today. This is the third day of our summer school 2021. We will start in few minutes, and so we will let the time to the other students and attendants to join us. Okay, so we are on time, so it's time to start. And I'm Francesca Boccafoschi from UPO in Italy, and I would like to share my screen with you to start with the cultural presentation. Okay, here we go. So just to start, and I would like to give you few ideas of what you're gonna see coming in Italy or coming into our region. Because actually, I think that Italy is pretty well known for the history, for the art, and for food, for instance. So I would like to grab your hands and stay with some minutes with me just to going around Italy, starting from a video that I would like to share with you, which is a just 1 minute video, but you can see the real panoramas that you can see when you are here in Italy. And that's it. So just few images to come very quickly here in Italy. You have seen our sea, our coast, our mountains, our food and wine, of course. But just to give you another idea of something that is culturally Italian, is our the popular gestures that we use. Every time that I go abroad and I meet friends from other countries, they are always laughing about the way we used to speak, because we use our hands a lot. In fact, in the last year and a half, with the remote classes and remote meeting and whatever, it was pretty hard for us Italians to properly explain to the audience the ideas that we had. So have a look here. For instance, we use a lot of gestures, and we use it speaking, but even to give messages in silence, so we can understand each other even with let's say easy communication, and we use it a lot. And actually, yes, we have I think all over the world knows this, I don't care, but we don't use it that much actually. We use it more. Finito is finished. We use go away. We use I'm here alone. So, or stop it, or are you nuts? Yes, we use it this actually pretty a lot. Actually, we are using even other gestures that I think they are not really Italian, like this one, Italian. We do not use it, but we obtain this enrichment of our gestural language, I guess from the Anglophone. So this is not Italian. We have to be honest. This is more Anglophone, but we use it. So this is just a silly start, but actually it's something that if you ask around the world about Italians, apart of pizza and some other stuffs, they talk about our gestures. So yes, we use it a lot. And if you think about Italy, a part of the images that I showed you before, there are another important point, coffee. Yes, we love it. And actually we have two different ways to taste it. The mocha, which is the more, let's say, traditional one, and is the one that was used at home mainly, even if today we do not all the family still use the mocha coffee anymore, and the espresso. This is for sure the one more used all over the world. But here in Italy, having a coffee is not just something related to, let's say, food, but actually is a moment that we take a kind of break, but actually is even a way to keep in contact with friends. If you have not a lot of time, let's go and have a coffee. Coffee for us is just a few minutes. It's not a very, very long break. Just a few minutes, a short coffee, and that's it. But it's a way to get connected with friends and with colleagues too. Actually, we use it here at the university. We use it in lab, even with students. So when we have to stay all together and maybe discuss even results, well, yes, we have a coffee all together. And I have to be honest, the best ideas and the best project is they pretty often born in front of a coffee. So this is a very nice tradition that we have, and I think I know that it's pretty used all over the world, but it's another way to taste coffee here in Italy. Is something more not only familiar, but even, you know, for friends. So there is another point that actually is, I told you that we have two different kind of coffee, mocha and espresso, but have a look here. It happens sometimes that if you enter in a pub or in a restaurant or whatever, you can choose all of this different type or coffee. So you have espresso is the, let's say, the normal one, double, short, or a cappuccino, which is pretty much well known all over the world. Mochaccino, this is something actually not that common outside of Italy. Marocchino, which it seems like a smaller cappuccino, let's say. Anyway, so actually you can have fun to go around here in Italy and test coffee, but not just the normal one, but you're gonna choose a lot. And coffee is for sure related to our breakfast too. And that's it, here is our breakfast. I think that even this one is pretty well known, but what we have normally is a cappuccino with a brioche. The only point is that brioche actually is not only one kind, but you have a very huge possibility to choose whatever you want, with cream, with chocolate, with marmalade, with anything, so alone. And so it's pretty nice to enter in a bakery and choose the brioche that is perfect for that day. So this is another very nice tradition that we have here in Italy. And yes, actually, a part of coffee and breakfast, if you move along along Italy, you have the opportunity to taste so many different kind of food, which are related not to the whole Italy, but actually they are related to regions. So this is something that may be weird, but actually we have very, very strong regional traditions. So for instance, if you would like to taste a very good arancino is from Sicily. So it's from this island here in the south. If you would like to have an arancino in the north, it's not a good idea. It's not the real one, you know, because actually Italian, they are really proud not only to be Italian, but even to be of a part, so of a specific region in Italy. So if you would like to taste the real arancino, you have to go in Sicily. Or for instance, if you would like to taste, I don't know, risotto alla milanese, of course you have to go in Milano. No way. So this is nice, but actually it's something that is fascinating, not only me, but I think it's fascinating the tourism here in Italy, because even if you move just for a few kilometers from one area to another one, you can find very, very different traditions and very, very different food traditions and other. What you see here in the middle of this slide is the region where I'm located now, where UPO is located, which is Piemonte. And Piemonte is for sure well known for cheese, for chocolate, in Torino in particular, and of course for wine. Have a look on the right. Here, what you're gonna see is simplified map of all the wine that are produced only in our region. So the reason why Italy is so rich in food tradition is actually because we have from the mountain to the sea, all the different temperature and environment. So this is one of the secrets that is behind the richness of the tradition here in Italy. So, but let's go farther. So, but let's stay here in Piemonte. So what I would like to show you is, oh, sorry, no, I was forgetting this. There is another point. The other point is that actually a part of food, even dialects are really, really different, moving from a region to the other. And the reason why our language, I mean, we use Italian, of course, but if you move in all the regions, we have dialects, and dialects are really different, above all from north to south, but even from east to west. So, and in some cases, they are really languages, so even grammar is different. Of course, we do not speak all the Italian dialect. Sometimes you know one or two of them, and actually going on with the generation, we are losing a little bit this tradition. So we are not using dialect anymore, but we have an accent. The accent that is very, very different from the north to the south and from the east to the west. So, and it's pretty nice to that we are able to understand if one person is from Naples or from Milano or from Rome or whatever. So this is, for us, is pretty easy to understand. I'm not sure is that easy for people coming from other countries. So, but apart of this, I would like to stay here in Piemonte, so in our region where UPO is located. I would like to give you some information about our university. And our university is located in 3 different cities in the same region, in Piemonte, Novara, Vercelli, and Alessandria. Novara is the city where I'm located normally and even now. This is something that is really nice because actually, as I told you before, is a way to enrich the tradition and even the possibility for students to interact. In the whole region. so to have even have different experiences, even in terms of working experiences. Actually, the three cities are they are dedicated to specific faculties. So for instance, here you're going to see that in Vercelli, we do have, let's say, the main building, which is the rectorate. And the study areas are humanities, material science, biology, and nursing. For instance, in Novara, actually, we do have we are more balanced on economic and medicine, but actually even law is moving from Alessandria. That was the first base of law in UPO, is moving even here. And so Alessandria, the third city, is we have political and social sciences, chemistry, biology, law, and whatever. They are not that far. For instance, in 10 minutes by train, you can go from Novara to Vercelli. So this is, if you are used to live in a bigger town, for instance, in Rome, it makes no sense, because in 10 minutes in Rome, you are moving from, I don't know, from one metro stop to the second one, more or less. But here, cities are smaller, of course. So UPO is growing pretty fast. Actually, these numbers are not the last one. They're not updating. Right now, we are 15,000 more or less students. And actually, we are divided in departments. And in all cases, all our faculties or our departments are very, very well oriented to the international part of the university. So we have Erasmus programs, and we have even other international programs that open UPO and welcome in UPO international students. In fact, we do have international study programs already with several universities, and still we are signing other agreements with other universities, for instance, even in Canada and pretty far from here, let's say. In order to welcome the students, Italian and international students, of course, UPO offers different services, so orientation, cultural service, financial service, and whatever. And just to close this first session together, again, as I did at the beginning of this talk, I would like to grab your hands again and have a walk in UPO. And that's it. So the cultural session stop here. So I give the stage to our chair.
Thank you, Francesca. That was a wonderful tour through Novara. I must say, I was waiting for the hazelnuts. So I think that's one of, there are so many wonderful things to do and eat and drink in Novara. So absolutely, I think it was just a small taste of what visitors can do. Thank you very much for that. I'm sure our Mexican, British, and all around the world students are now looking forward to a visit to Novara. Thanks. Okay, so we're going to start with the scientific sessions. And the first session of today is by Dr. Simone Merlin. Dr. Merlin is an assistant professor at UPO. He holds a master's degree in pharmaceutical and medical biotechnologies and a PhD in biotechnologies for human health. Since 2007, he has been working on cell and gene therapy field, especially for the treatment of hemophilia A. His research is focused on the identification of the cell types involved in FP3 production and the clarification of the main cell types and the molecular mechanisms involved in F8 immune responses. Thank you very much, Simone.
Thank you. Thank you for the presentation. So today I had this idea to share my screen with you to present something that maybe young scientists can be interested in, and this is something we used for gene therapy approaches, actually, which are viral vectors for gene therapy. So when we talk about gene therapy, we for sure not talking about this kind of gene therapy. And for sure, what we see on the screen in Hollywood movies with these measles virus able to cure cancer, that 3 years later give us these results, is not gene therapy, or at least not the output of this treatment, let's say. For the gene therapy, we talk about the cells. So the cells which have any kind of cells has an instruction manual to follow in order to do a particular specific job. This instruction manual is actually 10,000 pages, 20,000 pages, and those pages are genes. So you have to imagine that a single letter error, so a single error in a letter, just a change of a letter in a single page, is changing the meaning of the entire page. That is a genetic disease, okay? And what gene therapy want to do is correct that disease by adding a new correct page. So gene therapy has the aim to deliver genes as drugs, as therapeutics. It can do it by delivering the new functional genes, both ex vivo or in vivo, in order to correct a genetic deficiency. The ideal treatment is a single treatment which has long-term and durable effects. For doing this, we can use actually both viral and not viral vectors, but something that in nature is really able to do this kind of this kind of job means moving from a cell to another, moving genetic materials, is are viruses. So we want to take something that is very often is bad, like a virus, and transforming into viral vectors. So we can remove whatever are the pathogenic genes, whatever is causing the disease from the virus, taking only the goods of these viruses, and create viral vectors which allow us to carry, to transfer the correct gene into the desired cells. So we can use different viruses to create different viral vectors, which at the end have common key properties, which are safety. Of course, we want to something that is biological compatible and is not damaging at the end the recipient. Low toxicity. Of course, these viral vectors created should not damage the infected or transduced cells. Stability. Very often pathogenic viruses have these characteristics that they tend to change their genome during time, and this is something we want to avoid, of course. You want a kind of cell type specificity, because maybe you need a particular cell type to produce a particular protein, and you can exploit different techniques in order to achieve this goal. Last is the identification. This is something that is added in order to identify, to verify the positioning of the viral vector, especially if you are considering an in vivo gene therapy. So it means that you are injecting these viral vectors into a human being. You can identify, basically, you can identify these vectors and better to improve safety. Sometimes this identification is also a particular gene that can be used in some cases as plan B to kill the cells that received the gene in the wrong way. So with all this in mind, you create your special Trojan horses, which are perfectly shaped for the delivery of the correct gene. What kind of viral vectors are we using? So in the picture here, you can see adenoviral vector, lentiviral vector, AAV, or adeno-associated viral vector, retroviral vector. These are the most common, the most famous used viral vector. But you can even exploit properties of other viruses, such as herpes viruses, measles virus, and so on. All of these viral vectors have different features that can be used for the gene therapy purposes. Of course, even though we pay attention in the viral vector construction, we still have some risks or side effects following the gene therapy. First of all is the immune reaction. You have to think two things. One is we are injecting a viral vectors, so the immune system can react against these virus. The other risk is the immune reaction against the therapeutic gene. So our body can react against the protein that actually was never produced by the body, right? The other great risk, this regards basically the viral vectors that are able to integrate within the genome of the host, is the insertion of mutagenesis. Basically, this happens when the viral vector transfer the gene in a position that is able to transactivate other genes, something like proto-oncogenes that are able to cause damages, something like cancer or other deleterious side effects. Another risk is the idea of targeting, because maybe we want to avoid the expression in certain cell type, or we want to avoid the insertion of a particular gene in a particular region of the genome. And of course, still related to the integrating viral vectors, there is a gene inactivation. So you have basically the integration of your new gene into an active gene that at that point is inactivated. Of course, we design our viral vectors in order to reduce as much as possible these risks and side effects. And how can we do that? We can control our viral vectors in different ways. Let me take as an example the liver, and here in the central part you see a liver sinusoid. So basically what we can do is control the viral vector at the entry level by changing the envelope, basically the suite of the vector. So by changing this cover, we're changing its ability to recognize or be recognized by different cell type, interact with different cell types. And this way we can reduce the number of targeted cells. Another way, another layer of control is using a tissue-specific promoter. So if basically if the coating, if changing envelope is not enough, we can add a sequence that is active only in particular cell types. This way we can try to avoid the transgene expression in unwanted cell type. As last, we can also add an additional control, which is working at post-transcriptional level, which is called MIR-T, or microRNA target sequence, which is a sequence that is completely complementary to microRNA, which are produced by cells. Every cell produces this different type of microRNA, and these microRNAs are able to bind a particular sequence and stop the translation of an mRNA. In this way, we can avoid the transgene expression in unwanted cell type. So by controlling the entry, the transcription, and the post-transcription, we reduce a lot the not specific transgene expression, right? So in our lab, we're working mainly on hemophilia gene therapy. And so this is something that I want to share with you, just an example of what you are doing. So hemophilia A is an X-linked bleeding disease. This is affecting 1 every 5,000 males. It is caused by mutations or deletion of factor VIII genes. So if you remember, just a single letter in a page, so the factor VIII page is causing this disease. And according to the error, this disease can be mild, moderate, or severe. So according to the residual factor VIII activity. Patients are treated affected by this disease are treated with a recombinant or plasma-derived factor VIII. There is not a definitive cure. They are treated with the replacement therapy. So basically, they received what they're missing. And this factor VIII is a very important cofactor in the coagulation cascade. So hemophilia A patients do not clot correctly. The only problem of the recombinant, so the recombinant plasma-derived factor VIII is that the administration of these substitute is causing the formation of antibodies that neutralize the activity of infused factor VIII in about 1/3 of the patients. So this is a big issue to overcome. Since it is a monogenic disease, hemophilia A is indeed a good candidate for the gene therapy. And from our past cell therapy studies, we discovered that liver sinusoidal endothelial cells, so which are the endothelial cells that are lining the sinusoids, like the scheme, like in the scheme I showed you before, they are the main source of factor VIII in the body, within the body. Another good source of factor VIII are hematopoietic cells, so the one that are coming from the bloodstream and from bone marrow. So our idea was to both with our lentiviral vector construct characterize and target the factor VIII producing cells. So what we did actually, we studied this vector before in vitro and then in vivo. When we used basically the factor VIII promoter to control the transgene expression. This is a transgene that is called GFP, that is regularly produced in nature. It's produced by jellyfish, Equora victoria, and we use it in lab as a marker gene. When we injected our lentiviral vector, we observed that both endothelial cells in the liver and hematopoietic cells in the spleen were able to express the transgene, meaning that this promoter was active in these two cell types. And this was confirming what we observed with our cell therapy studies. So we removed the GFP and we substituted with factor VIII gene. And what we obtained in these mice was a correction of the bleeding disease without antibodies formation. So with no immune reaction against the transgene. Okay, so if you're more interested in this topic or you're interested in this particular work, you can find the paper on PubMed. So of course, this was just a taste of what are viral vectors. It is a real huge word. One of the use of this viral vector is not only the gene transfer, both in vitro and ex vivo or in vivo for the gene therapy. You can even use it for vaccines, for example. AstraZeneca vaccine is one of these examples. And right now, the researchers are still trying to improve these vectors by increasing safety, efficiency, specificity, efficacy, they're performing more immunological studies in order to understand what are the immune reaction against both the transgene and viral vectors. New studies are trying to use particular systems borrowed from bacteria, actually, for not only add a new page, a new correct page, but to correct directly that letter that is wrong in that particular page in our genome. Okay, in order to correct our own genome. It's called what we call gene editing. And there are a lot of studies ongoing, and I'm pretty sure that very soon we will have more and more results on these topics. I'd like to thank you for your attention. This is our great, wonderful lab, and thank you again for your attention.
Thank you very much. That has a very, very interesting talk. Can I also remind the students and all attendees, if you have any question, please post them in the chat. You can do it through the Facebook or the YouTube chats as well. So do let us know if there are any questions. Well, we just wait to see if there's anyone asking. Can I just ask, of course, it was very interesting. How far is this from the clinic? What are the challenges before it goes? You did mention safety, for example, and I cannot help but think that even with a lot of research, there might be some side effects that we're not encountering preclinical or clinical trials. What would you think will be the main challenge to take this to the clinic?
It depends on the most challenging thing is answer your question, actually. No, there are, it's not, the answer is not straightforward. I have to be honest. There are already clinical trials ongoing with the AAV, for example, the adeno-associated viral vectors for Hemophilia A. Those are the last one. Hemophilia B before. So it's just because the transgene was shorter and was giving lower problems in viral vector production. There are also some clinical trials for retinal disease ongoing, always with the AAV. Babies after gene therapy ex vivo for ADAS-Kid are still under control, but they seems to be cured. They were basically babies with an immunodeficiency now they can run away outside, so they can be happy now. So it's getting harder because the safety first. So and this is always the biggest concern. You have to find the best way to use a particular vector against another one. If you talk with people that are working with viral vectors, their vector is the best one. If you talk with the AAV person, they are in love with the AAV and they will prefer it among the other. In my opinion, according to what you want to cure, what is the disease, what is the gene you have to deliver, whatever is your target, you choose your viral vector. Then, of course, you can still improve a lot Lentiviral vectors are, for example, we are using integrating vectors. So these are lentiviral vectors. One of the most frequent issues is the insertion of mutagenesis. Okay, now they are modifying these vectors in order to let them integrate into particular zones called safe harbor loci in order to not damage the host. So we are they're there, but not as many as they deserve. That's the answer.
We've got a quick question before we have to move to the next speaker.
Okay.
From Neda Shahedi. How effective is gene therapy in treating hemophilia?
Hemophilia A in general it seems to be effective for us. So the clinical trial for hemophilia B is running since a long time now, and even though the factor IX production is low, it's still there, and the patients are very happy. With the factor VIII, it was kind of harder, but we have now data up to 3 or 4 years, if I don't make a mistake, following gene therapy. They were aiming at 5%. Some of the patients reached 100%. Other clinical trial, this is the very first clinical trial that was from Biomarini, if I don't make a mistake. Other clinical trial are updating every some month with the results, and they're showing very good results, even though they are not reaching the high levels reached by the very first one.
Thank you very much for answering the questions, and I'm sure that all delegates are joining me in thank you for your time and the talk. Now we are moving on to the next speaker. We will start sharing the screen. Excellent, everything is ready. Our next speaker is Dr. Care of Rasotti. Over the past 20 years, Dr. Rasotti has gained deep knowledge on the biology of hematopoietic stem cells on the immune system responses in both physiological and pathological settings. In particular, she began her studies during her master's internship testing in vitro the survival of leukemic HSC to different drugs. For 2 years, she was a graduate research assistant at the Memorial Sloan Kettering Cancer Center at New York, America, where she studied the role of T-cell cytolytic pathways in bone marrow transplantation. Then, as a PhD student at the Università Vite Salute San Raffaele in Milan, she characterized the human adaptive immune system development in a humanized mouse model. As a postdoctoral fellow, Dr. Rasotti joined the Columbia Center for Translational Immunology in New York, where she was involved in projects aimed to develop humanized mouse models for studying autoimmune diseases. She is currently working in the histology laboratory at UPO on projects focusing on the identification of cells and mechanisms involved in the immune response to factor VIII in the treatment of hemophilia A, with the aim to promote its tolerance. She has authored 24 papers on international journals with editorial board. Welcome. Thank you.
Hello, everybody, and thank you for the nice introduction. So today I'm actually going to share with you just a few words on models for studying type 1 diabetes in mice, and in particular, how to study that really using human cells that you can transfer in the mice. That's why it's called Humanized Mice, for modeling type 1 diabetes. So just a few words for the introduction of type 1 diabetes. It's an autoimmune disorder involving the specific destruction of insulin-producing pancreatic beta cells. And it's mainly due to a combination of a genetic predisposition and also environmental factors. And there are also some which are called stochastic events, which mainly means that we still don't know what they are, but they led to the total destruction of the insulin producing beta cells in the pancreas. And actually, what is now very well known is that you become diabetes, so you get a diagnosis with diabetes when more than 50% of beta cells are already being destroyed. So the first destruction of the beta cell starts and the person is not aware, and it's really a very specific autoimmune reaction. And only when most of the beta cells are destroyed, then you are not able anymore to respond correctly to the glucose. I just want to quickly also remind you that type 1 diabetes is totally different to the type 2, which is a metabolic disorder mainly, not an autoimmune disease. So, and while type 1 diabetes is really an autoimmune disease, type 2 is totally different in terms of pathophysiology. So it's a very complex slide, but when we go to immunology, that's what normally immunology likes to show. But the key point is that when you have type 1 diabetes, you really have what is called a loss of tolerance against the insulin and the beta cell specific antigens, which means that what is normally should be self for the organism is now not self anymore. So our own cells, in this case, in case of the type 1 diabetes patient, is attacking the beta cells. And is mainly for mediator a T cell mediated, which means you really have the CD8 T cells attacking the beta cells and destroy them very specifically. For example, you don't have any problem with the alpha cells, which are present in the islet pancreatic cells there. And you still find also autoantibodies against the specific antigen like insulin, although the B cells are mainly involved probably with the antigen presenting cells, more than have a direct effect due to the antibodies production. And in terms of really the effects in the immune regulatory mechanism, there are many studies showing that these patients have a defective in antigen presenting cells. They have normal expansion of autoreactive T cells and defective regulatory T cells. So these all things all over, they are making easier the reaction against the beta cells in type 1 diabetes patient. So, so far, what is possible is to study autoimmunity of type 1 diabetes in some animal models. There are spontaneous ones, like for humans, you have mice and rats, which we know they develop type 1 diabetes, or you can induce with a very, very difficult kind of experiment, the type 1 diabetes in the models, in mice mainly. While in humans, you can mainly study the circulating T cells in the peripheral blood of the patients because they are easily accessible, and the pancreas, but only, of course, after the death of the patients. So there are many limitations in these studies because most of the time, first, as I mentioned, you can only study from humans the circulating T cells and nothing of what is happening really in the pancreas. And often you study the patient when already the disease has appeared and has almost you have almost the destruction of all beta cells, which means you really don't see the first stages. As I mentioned at the beginning, the first, when the reaction against the beta cell starts, the person is not aware and doesn't have any signs, and only later it will develop really the resistance to glucose. So ideally, if you want to study the type 1 diabetes, the modeling in vitro or even in vivo, the key cells that are important are for sure the beta cells, okay, which are the cells that are normally destroyed, and then the component of the immune cells. And for sure, I mentioned one of the most important are the T cells, and in particular, if you think it's a cytotoxic response against the beta cells, are the CD8. And then in these pictures, you see that there are, of course, many other factors that are players like cytokines and the famous stochastic events. And ideally, if you get the nice model, then you can study possible therapies, drugs that can be used, and the dream of medicine, personalized treatments. So how can we do that? Nowadays, one big application, of course, is the possibility to differentiate in vitro specific cells, starting either from embryonic stem cells or more recently from what are the inducing pluripotent stem cells. I saw that you had a presentation on Monday on the different stem cells in regenerative medicine. And so I don't have the time here to describe specifically these stem cells, but the embryonic stem cells are the one you normally get from a developing embryo, very early stages, which they will give rise to all the tissue in the bodies. So they have a lot of potential. But nowadays, also many labs are working what are called inducing pluripotent stem cells, which basically have the advantage that you can get what is already a mature cells from an adult, and you can de-differentiate it and bring him back to the stem cell-like capability. So if you're able to get these cells in vitro, which nowadays you can, there are now very well described protocols for which really you can culture and differentiate the cells in specific tissue. And in this particular paper, what they were able to do is to differentiate these cells in the beta cells, or just progenitors just before beta cells. And the dream, of course, is to be able to then transplant these cells back in the patient to cure diabetes. Although that will be not enough, because the patient will still react against these beta cells, because the autoimmunity does not go away. So in terms of then reaching really the treatment of the patient, we are still not close to that. But the good thing is that nowadays it's possible, definitely, there are here a list of papers which they show that it's possible to get the beta cells differentiated from really samples taken from type 1 diabetic patient, most of the time fibroblasts, which are taken from a relatively easy biopsy from the skin. And more recently also, it was possible to having inducing pluripotent stem cell from peripheral blood of the patient and then differentiated in beta cells. And I just want to mention then, in terms of studying in vitro, this group actually was able, from the same patient, to obtain the inducing pluripotent stem cells and differentiated them in different tissue, including the beta cells. And taking the T cells and the immune cells from the peripheral blood, it was able to testing in vitro some of the reaction. But, okay, the title was Humanized Mice. So why that? Because ideally, if you can test in an in vivo model the action of this specific reaction and between mainly the T and B cells and the beta cells, it would be nice. We know that mice in general are widely used because they're relatively cheap and they breed very well and they occupy relatively small space. And the word humanized mouse is probably known from many of you, and it refers to a mouse carrying functional different parts of humans, can be even on the genes. But for this kind of studies, you really need to transfer human cells or tissues in the mouse in order to be able to study there what's happening to those cells. And so I just mentioned before that they were able to differentiate the inducing pluripotent stem cells in beta cells. And in order to study their function, what they did, they transfer now these beta cells under the kidney capsule of an immunodeficient mouse. That's an important thing. If you are transferring cells of human origin in a mouse, you need to have a mouse that will not react against the human cells you're transferring. So you use immunodeficient mice. And you use the kidney capsule because it's an easy place, relatively easy, where you can put the tissue and will easily be vascularized and also will remain there. And I just want to point the attention to this graph. in which they show basically each number represents a different mouse that was transplanted with these differentiated beta cells. And they tested what was happening if you were giving glucose to the mouse. So 0, which is the black bars, means that was not challenged yet. So it's the basal level human insulin production. Human means that really it's coming from the beta cells you transfer, because you're specifically testing the human insulin, not the mouse one. And then after you give the glucose, of course, you expect the insulin to go up. And for certain mice, if you look at the black bar compared to the white, it's true, which means that the beta cells are functioning properly in the mouse then. But for some, it's not true. Okay, for example, if you look at mouse 2, you have actually a decrease, which means it's not enough to have a differentiated beta cells in vitro. You need to test if it's functional in vivo, in this case, then in this model of mouse. And this was possible also for comparing beta cells that were differentiated either from type 1 diabetic patient, the one highlighted here as type 1D, or in the one coming from non-diabetic people. And what this is showing, the principle is the same. They were differentiated in vitro and transferred under the kidney capsule, is that there is no difference in the production of insulin between mice which receive the beta cells differentiated from type 1 diabetic patient or the one from normal, which is not surprising. That does not surprise us, because the problem with type 1 diabetes is not in the insulin production, it's really in the autoimmunity. So then how do we study the autoimmunity part? Well, we need to complete the humanized mouse. And so we need to add the human immune system there, which is actually how the whole story, most of the story from humanized mouse started actually. So the idea to be able to study hematopoietic stem cells of human origin developing in a mouse, with the advantage to be able to really study the first development of the human immune system from the beginning. And with the advantage also that in mice, you can easily access organs that in human you cannot. You can take bone marrow, spleen, for sure. And the way to obtain a human immune system in a mouse, again, the principle always need an immunodeficient mouse strain. You see there a list of them. It's not important. You just mainly need to know that they cannot basically reject the human cells you are basically transplanting. And in order to have the human immune cells, you need to have a source of hematopoietic stem cells. And there is a list. So you have fetal liver, cord blood, even peripheral blood, or adult bone marrow. And especially if you want to study the reaction that is happening during the development of type 1 diabetes, one possibility is really to use the bone marrow samples coming from a type 1 diabetic donor. And of course, as a control, then you will have mice which are going to be generated with healthy control hematopoietic stem cells. And this model, which use really cells, hematopoietic stem cells coming from the bone marrow patient, is called Personalizing Immune Mouse model, because it's really somehow the mouse is going to develop the human system of that specific person. It's a complex because it requires a lot of passages. You need to get the sample from a human bone marrow, and then you get the mice transplanted them. You need to wait for the human immune system to develop. So normally it takes at least 6-8 weeks to have a mature human immune system there. But it's possible, and already in this paper that is mentioned here, they show that if you transplant hematopoietic stem cells coming from a type 1 diabetic donor, you already can observe in vivo a kind of alteration of the T cells, which are basically normally acquiring without any kind of stimulus, but they are acquiring a more active phenotype, which means the threshold of activation is higher than for the healthy controls. So what will be the ideal steps now is to combine in vivo the transferring or the bone marrow hematopoietic stem cells of a type 1 diabetic patients with the transfer of beta cells, which are going to be instead differentiated from inducing pluripotent stem cells of the same, of course, patient, because then they are autologous. You can really study the interaction there in the mouse. If it's not possible to get both the hematopoietic stem cells and the samples for getting beta cells, what you can think, but is still not now demonstrated, is to get even the hematopoietic stem cells from the inducing pluripotent stem cells, which apparently is more difficult. There are many protocols, but they are not still functioning very well compared to the one that gives rise to beta cells. Then, of course, since you never will have the perfect model, you will still miss one of the more important things, which are if you study in a humanized mouse, the environmental factors. But let's start to, and we are working on that as a scientific community in getting already a model in which you can have both the human part, the human immune part, and the beta cells, and then we can then provide a move to the other. And with this, I have finished my presentation, so I'm happy if there are any questions to answer.
Thank you very much, Chiara. Very interesting presentation. And while we wait to see if there are any questions coming, can I just ask, of course, the humanized model looks amazing because there are many constraints when you are using an animal model, but you want to translate that, of course, for humans. What is the difference in cost to have a humanized model to a normal rodent model? Because I imagine that these are especially bred animals.
Well, okay, so the basic is to have an immunodeficient mouse, which is normally a little more expensive also to keep because you need to keep in a special environment because it can easily get infected. So you need a very specific system. You need to work under a hood when you manipulate them. So they are a little bit more expensive on that compared to other mice you're using. And then in order to humanize them, you need to think all the different tissue you need, how if you need to get hematopoietic stem cells, you need all to process them. So they are definitely a little more expensive than a normal model. That's for sure.
Thank you. That's very interesting to know what are the actual technical differences and how you keep them and the processes. Of course, this was for diabetes type 1. What would be the difference, or how could you apply your model for diabetes type 2? Is it anything you can do? Okay, so there you can think for sure the fact that you can get beta cells from inducing pluripotent stem cells can definitely then be translated to the study of type 2 diabetes. There you don't need anymore the immune part because it's not an autoimmune disease, the type 2. There, most likely what you will need is, of course, to get these beta cells from patients that develop type 2 diabetes, because then the defect will be probably more on the beta cells part than the immune system. I show you the slides where if you compare the beta cells function that you obtain either for type 1 diabetic or non-diabetic is basically the same. There are no problem in insulin secretion because it's not a problem of beta cells. Probably if you do this with type 2 diabetic patient, maybe you already get some problem there because it's an intrinsic defect probably of the beta cells. So you can think you can translate in this way. Thank you very much. I think there are no more questions from the channels. So with that, we thank you for your talk. And we are going to have a brief break, and we are going to see everybody back at 4:25, please. UK time, 5:25, Italy time, and 10:25 in Mexico. So thank you very much, and we'll see you in a couple of minutes.
Thank you.